Hello all. In this lecture, in this week, we will cover indoor air pollution monitoring for biological samples, for example, bioaerosols. We will cover the sampling methods, analysis methods, and cover some instrumentations. So, introduction I will give, although we have seen this, that bioaerosols are suspensions of airborne particles that contain living organisms or are derived from living organisms. So these could be what? These could be viruses, bacteria, fungi, pollen, plant and animal debris, and fragments and products of these. Now these bioaerosols can be dispersed by attaching to other biological or non-biological particles. These could be soil, dust, skin flakes, saliva and water droplets. So anything these bioaerosols are once emitted, they may, for example, soil, if they are attached to the soil and when the soil is resuspended, or they are attached to the dust and the dust is suspended, and then humans will inhale that and our exposure will happen. Skin flakes is shedding from the human themselves, same for saliva. So then this makes the bioaerosol amenable for transmission to other people. And if they are in water droplets, when the water, for example, is coming out of a shower or a tap, there may be small water aerosols as well, and together with them the biotic material will come in the suspension and then we may be exposed to that. As for particles, the bioaerosol sizes also range from 1 nanometer to 100 micrometer even. And these bioaerosols are important because, as we know, they can cause many adverse health effects, which could be acute, meaning immediate exposure and harmful effect, or chronic, which means low dose exposure over a long time, which creates a health effect. In acute, we are exposed to some high concentration and immediately or almost immediately we get some harmful impact, or in chronic we get small doses over long period of time, and then over time we have some harmful health effect. So what are the sources of the bioaerosols in the indoor environment? The first, of course, is humans. So we are always there moving around or emitting some bioaerosols. So the bioaerosols will be there in the circulation and in the living spaces of the inhabitants. Then bioaerosols may also be outdoors, and then from outdoors they can come inside through openings which exist in doors and windows. So as soon as we open a door or a window, when the air is coming in, together with the air we will have bioaerosols coming in. Then air conditioning systems as well. So when the air is being sucked in in these systems, we will have the bacteria coming inside. Or maybe in the parts of the AC system, we have dust and microbes attached to the parts of the air condition, for example, where the flaps are. So when the AC blows, then we get bacterial or microbial exposure. Although if we are looking about the flaps, they are still inside. For outdoors, it's the air coming from outside through the air conditioning system, which will bring the aerosols from the outside. We may have water and moist areas. For example, when we have leaky pipes or joints in the plumbing system, those moist conditions will sort of proliferate the growth of microbial entities, and then those can somehow become part of the indoor environment. Then in bathrooms we use shower, and since the environment is damp and moist, there will be mold or bacterial or microbial growth. So that's one place. And then if the water is coming through the shower, then again we have aerosolization. In kitchen, if we have stale food, or the counter or the tops have not been cleaned for some time, then bacterial growth may happen or microbial growth will happen, and then we will be exposed to those bioaerosols. And also in other damp areas, wherever they may be, even if they are small pockets, we will have bioaerosols. Then there are several other sources, for example pets, dust. So pets will of course release their own biotic material. Dust itself may have some microbes attached to it. If there are healthcare facilities, then we may have some more infectious bacteria or microbes be around. And plants which may release pollens or other living or living entities or entities derived from the living parts. So since we have so many wide variety of aerosols, we also have a wide variety of sampling methods. A wide variety of these methods are available, but there is a bit challenge because for many of them the standard protocols are still being established. Broadly speaking, as in the chemical sampling, here also the sampling can be defined as passive sampling or active sampling. Now, passive sampling is a non-qualitative approach. Well, it can be used quantitatively based on the gravitational settling of particles on a collecting surface. So it can be just like a plate and the things are depositing on the plate, and then we do some counting. In this particular method, so the surface of the collection plate will act as a source of bioparticles. In some instances, probably also a source for bioparticles, reflecting their air concentrations averaged over the time period of sampling. So if we make the collection for one day, then whatever we get is sort of a reflection of the air concentrations or air contamination over that one day period. So surface acts as sources and sinks for bioparticles, reflecting their air concentrations averaged over the time period since the surface was cleaned. Sampling is basically based on the exposure of agar plates and agar coated slides in the air. So we will put them and then let the bioparticles sort of settle on them, and then we analyze these plates. What is done is, nutrient medium is given, the proper incubation temperature is given, and the growth of viable biological particles is observed and is expressed as colony forming units. So one can visually inspect these colony forming units per meter cube of air or do other analysis. In general, the Cox sedimentation method is commonly used. In this method, the sample is usually collected at a height from the ground, about 1 meter above the ground, and suitable care is given in the placement and removal to prevent contamination. So we will keep it in a relevant place in the indoor environment, but not in a place where a person will manually put something, or it gets disturbed by people moving around and throwing things, and those extra things go in the plate and they basically skew the results. So suitable care has to be taken in the placement and the removal so that the contamination can be prevented. The collected plates, so once the sampling is over 1 day or 1 hour or 1 week, the plates are taken and are incubated for 24 hours at 37 degrees Celsius for aerobic bacteria, and for 3 days at 22 Celsius for saprophytic bacteria. Basically, we provide the ideal conditions for the growth of their colonies, because at the end of the day we want to know how many colony forming units are present. So we provide sufficient or the correct conditions under which these bacteria can grow. For molds, so we had 27, 37 and 22. For molds, the incubation temperature is 10 to 50 Celsius and for 1 to 2 weeks. So more time is given for molds to make colonies or for molds to grow compared to, for example, bacteria. Passive sampling is done for bacteria and these molds. It is usually or generally not recommended when sampling for fungal spores, because the fungal spores will possibly remain suspended in the air for a very long time or indefinitely. The whole method of this passive sampling relies on the microbial matter coming and depositing on the surface, but fungal spores actually remain suspended for a long time. So the method for deposition does not work very well when fungal spores are of interest, and therefore this method is not recommended. So mainly this method is used to sample particulates, which we know may settle down over a period of time, and bacteria, which also depending on the size fraction will settle. So it's mainly used to sample particulates and bacteria for research studies or any epidemiological investigations. The advantage of such passive sampling is that it is passive in nature. We keep the plate, we take the plate and we just put it, and if we put it in a secure environment, then we don't need to worry about it. We can just leave it without any electricity and go, and then come back and collect the plate. So the advantage is that it is simplistic, because it does not require any mechanical instruments. We just put it somewhere in a proper place and then take it back. Put the plate and then take the plate back. The method is economical for conducting sampling for long periods, because we can keep the plate for as long as we want. We do not need to come every day. We do not need to send everyone every day, and we do not need to add anything. We can just prepare our medium on the plate and let it be there. So it is economical. You go put and then forget about it, and you go and collect it after sampling and do your analysis. So it is economical for sampling for long periods. Combining this passive sampling approach with other air samples can also allow us to understand or characterize sources of airborne microorganisms with their relative contributions. So we can use plate techniques or these passive sampling techniques and other techniques and combine the information to even better characterize what are the sources and how important they are with respect to one another. Passive sampling limitations are that the method provides a snapshot of collected number of aerosols without relating to their concentration in the given volume of air. So we basically put the plate and do the collection for 1 day or 1 week, and then analyze it after 1 day or 1 week. So this method provides the snapshot of the collected number of bioaerosols, and we are calculating the colonies. So it does not necessarily relate the measurements or observations to the concentration of bioaerosols in the given volume of air. Also, when we have air currents and other disturbances in the sampling area, we can affect the count or even the survival of the microorganisms. So, in general, if we have air currents and disturbances, the chances of survival decreases. And when we have air currents happening, we can have less survival of microorganisms if the velocity of the current is high. The method gives an underestimate because the coarser particles are easily collected and the smaller particles are not that easily collected. So basically we will have better representation of the coarser particles. So if we want to know the microbial characteristics of a particular time, we may have to repeat the method several times. So one time, another time, and then check all these observations and say, okay, these microbes, they may be present. Doing only once may not always give the perfect answer in one go. Then we have active sampling methods. These methods rely on the external force for collection of airborne microorganisms on a culture medium. So we may have to draw the air using a mechanical or electrical means, for example using a pump, and then somehow using different mechanisms we collect the bioaerosols in culture medium. Different types of instruments are required depending on the mode of collection, the characteristics of the biological particle. For example, its shape, size, density and collection efficiency, and accordingly we may have to choose different instruments or the method, which these instruments or the means can be classified as impingers, impactors, filtration and precipitation based. So first we will look at the impaction methods. Impactors generally use an air pump to capture the air and rely on impaction of the sample into a petri dish which is containing our nutrient agar. So somehow the air is being sucked and we are relying that the air will impact the sample. So the bioaerosol will impact the sample onto the petri dish where we have our medium where they are supposed to attach to. So these airborne microbes along with other suspended material, along with other suspended particle, will settle by the force of vacuum. So they will be like sucked and they will attach to the medium. The airflow is controlled by slits or holes that will distribute the air evenly over the surface of the plates. So somehow we have even this is a plate and the holes are such that we have an even distribution of air over the plate, and the microbes will basically deposit onto the nutrient agar. Samplers which rely on impaction methods are often characterized by their inlet sizes, their shapes, collection chamber and the impaction surface. Now these surfaces can be solid, like a glass slide might be there, or semi-solid, which is the agar medium, or gelatinous. The flow rates used in such impaction devices are generally 10 to 700 liters per minute. Once the sample is collected, they are analyzed by culture based methods. We will look at these culture based methods in subsequent lectures. So these samples basically will have different sizes, shapes, collection chamber and impaction surface. Accordingly, they may be good for one type of bioaerosol, and depending on the flow rate we will optimize our collection efficiency, and whatever is collected will then be analyzed by one of the several methods available depending on what is the goal. There are several impactors such as spore trap air impactor, cascade impactor, slit sampler, stacked sieve 6-stage Anderson impactor, and rotorot sampler. These few samplers or these several samplers that are mentioned, they have been used quite substantially and sometimes are preferred because of their collection efficiency of various airborne microorganisms. So they have been evaluated a lot, and it has been suggested that these type of impactors have a good efficiency of collection of microorganisms, meaning they are less biased towards one or more, or not more biased towards another. So they fairly collect the different microorganisms so that our collected sample is a representation of whatever is in the air. In spore trap air sampling, we basically collect mold. So this is a commonly used impact sampling method for detection of mold in the indoor areas. Often this is referred to as spore trap cassette sampling. A sort of a cassette, collection cassette, has a slit opening. From this split opening, the air will pass and the particles will adhere to the surface of a sticky substance. Example could be triacetane. So the air passes through the slit and attaches to the surface of a sticky substance. So air is generally drawn through the cassette, and which is connected to a air sampling pump with a preset air sampling rate for a fixed time period. So we will say कि draw the air at whatever liters per minute for whatever amount of time, 5 liters per minute for 30 minutes for example. So any amount of sampling rate and periodic time sampling period can be fixed. Then after the collection is over, the cassette is resealed so that it does not receive any more biological material, and then is sent to the lab for further analysis. This is an example of a monitoring cassette, and this is our assembly. So as you see, this cassette is attached to the air and this pipe is attached to our pump. So the pump basically sucks the air from outside to inside, and whatever is in the ambient air is basically sucked in, and when this is happening, the spores, the mold will basically attach to the sticky surface right here. The cassette has a slit opening through which air passes and particles adhere to the surface of a sticky substance. So the particles will be sucked in, and in this time our relevant particles which were mold detection or spores, they will be attaching to the sticky surface. Then we will seal the cassette, cover the sides, the tops, and then send them for lab analysis. Spore trap air sampling is generally applicable for low to moderate loadings, and the results are underestimated for excessive loading because we only have a limited amount of surface area available for the biological material to attach. If we have too much biological material, all the sampling sites will be saturated. All the sites where the material has to stick will be saturated because we have too much material. So lot of the biological material will not get attached, and therefore the results will be underestimated. Therefore, generally this sampling method is applicable for low to moderate loadings. The sampling time usually varies between 1 and 10 minutes. Sampling time of 10 minutes is adopted in clean environments with little dust, because then we want to sample for more time so that we get appreciable amount of biological material that is stick to the sites. If we run only for 1 minute, we will not be able to obtain or collect enough biological material to do analysis. So we will run it for more time. If the environment is more contaminated, we will reduce the sampling duration. So sampling duration should be reduced based on the level of dust and expected mold spore levels. If there are a lot of dust or is a lot of spore possible, maybe from 10 minutes we will change the sampling time to only 1 minute, so that we do not saturate our available sites for the attachment. The next thing is the slit to agar impactor. The slit impaction sampler operates by rotating a culture plate below a long narrow slit or inlet. So here the collection substrate may be controlled to remain stationary, move continuously, or move in increments beneath the slit. An internal pump draws air to the slit at generally a flow rate of 50 liters per minute. So this is the internal pump drawing air through the slit, and the air is impacted onto 10 to 15 centimeter diameter plates with a nutrient agar. So this is our nutrient agar plate with the nutrient agar, which is kept here. The sampler is drawing the air, and our slit may be the culture plate which is below the slit may be kept rotating, so that may somehow enhance or improve the collection efficiency of the microbes. Sampling duration can range from anywhere between 1 minute to 60 minutes. Next are the multiple hole impactors. They usually operate by passing air through evenly spaced holes. Single stage impactor is most frequently chosen for viable microbe sampling. Another common impactor is our Anderson impactor, which has 6 vertically positioned sieves, and micro orifice uniform deposit impactor, which has 8 stage impactor with cut sizes down to 0.056 mm. So 56 micrometer is the cut size. In general, under each sieve, a petri dish with our nutrient agar is placed. So in all of them, we are basically using a nutrient agar plate where we are supposed to collect the microorganisms. So likewise in this multiple hole impactors also, a petri dish with nutrient agar is placed underneath each sieve. Now, as the air is passed through the consecutive sieves, the air speed increases, and it leads to a greater impact force when it collides with the sieves. So air speed increases as it passes through the consecutive sieves, causing greater impact force as the air collides with the sieves. Now the heaviest particle will settle on the initial sleeve. Immediately they will settle as soon as they enter. The lighter particles will probably pass through the first sieve. So lighter ones will travel along with the passing air, and they may be settled in the subsequent sieves. Particles of the bioaerosols, because of this, the particles of the bioaerosols are sorted according to their sizes, and then the colonies are derived from these particles of a particular size. So in this way, we can do our colony analysis by size of the sieve also. So we can relate those colonies to the sizes of the bioaerosols as well. This is the example. So this is our 6 stage. These are the sieves, and you can see they are numbered 1, 2, 3, 4, 5, 6. So they can be arranged in that order, and this is the our air. Impaction methods are usually advantageous because they allow to assess the ratio of particles which settle in the upper plates and those that settle in the lower plates, and to understand their influence on the respiratory system. Because when we separate these microbes by sizes, we know that the lower sizes are more likely to be inhaled by the humans. So if we have more colonies or more detection in the lower sieve sizes, we know that this particular air is potentially more harmful in terms of causing the respiratory health effects to us. So that's why these methods are advantageous, because they can tell us how much particles are in the upper plates which are coarser and how many are in the lower plates which are finer. Then examination of viruses can also be undertaken after we eliminate the other microbes by using compounds such as chloroform. So we will be able to remove or get rid of the influence of other microbes and just do the analysis of viruses. The use of impactors also allows the total sample to be collected on the same culture media. So we do not need any subsampling, or we do not need any, so to speak, extraction. There are some limitations, which is when we are using the semi-solid surface such as agar plates, there are limitations due to the overlap of collected microorganisms on one another. So maybe one gets collected over another. There may be also a possible decline in the collection, and this may be caused by the shock of a sudden collision with nutrient agar, and the possibility that the nutrient culture has overgrowth when we have a lot of air pollution. Now airstream also removes surface moisture, that means the surface stickiness may be reduced, and hence the ability to trap more particles may be also somewhat reduced. The performance of the sampler, which includes collection and the recovery, also varies with wind velocity. So we may need to maintain some optimum regime on the impaction velocity, the sampler's orientation, and the collection time also. So in summary, in this lecture, we have gone over the overview of various sampling methods, for example, passive and active samplers, this impaction based, and this is collection based, and these can be used for collection and further analysis of, once collected, the samples can be used for, the methods can be used for collecting samples and the further analysis. In the next lecture, we will cover more sampling methods.